Welcome back to our course, Machine Learning for High-Throughput Biomedical Data. In this lecture, we will talk about processing high-throughput data. Specifically, we will take one of the datasets from the publication we started to review and prepare it for analysis. In order to understand the samples and where they came from, we will learn about breast cancer classification and explore the associated NCBI bioproject. But before we start, it is important to note that in the context of machine learning, data selection and preparation are critical for good analysis. In fact, many data scientists and bioinformaticians spend the majority of their time cleaning, understanding, and preparing the data even before they get started. There is even a common saying, garbage in, garbage out. It means that whether your model or analysis are good or bad, if the source data is bad, there is nothing, not even the advanced methods and computational capabilities that will make your results any good. With that in mind, let's review the data we will be working with. In this lecture, we will be using the tBioInfo platform. We will use it to process raw data, so make sure you have the credentials you received with the course. If you don't have your credentials or have trouble signing in, please email me directly. The section we will use is under NGS, Next Generation Sequencing Data. It is called RNA-seq/CHIP, Parallel Analysis of NGS and Microarray Data. When using the platform, make sure that you take notice of the informational pop-ups that will appear while building the pipeline. These pop-ups provide detailed information on each step that we take. If you would like to learn about the algorithm, you can usually find the link to a reference publication in those pop-ups. So let's review the data set we are dealing with. The main topic that it pursues is precision medicine. This time we will talk about breast cancer and some of the treatments available for this disease. As we saw in the previous lecture, high-throughput data like genomics, transcriptomics, and others are becoming more and more affordable and accessible, while precision medicine relies on such information to diagnose patients, to understand disease at the molecular level, and even prescribe treatments to patients relying on such information. Let's take a look at what that exactly means. As you remember, the publication we selected deals with breast cancer. A mammogram is an X-ray picture of the breast that doctors use to look for early signs of breast cancer. In this example, you can see that the tumor is highlighted. Once it's found, it is critical to establish whether it's malignant or benign and what tissue it is originating from. Once the tumor is characterized by its phenotype, like size, tissue origin, etc., additional information can be obtained by checking for presence of specific proteins characteristic of cancer. If these proteins are known, immunohistochemistry or antibody staining can be used to find if they are expressed or not. Specific molecular markers are characteristic of cellular events such as proliferation or cell death. These are typically referred to as biomarkers. Based on these biomarkers, we can know something about the tumor and in many cases have a pretty good idea about treatment options. But protein expression, especially measured with immunohistochemistry, provides us with only a select number of biomarkers that we can check for. What if other processes are going on that we should take into consideration? Oftentimes, immunohistochemistry is used in combination with gene expression panels to explore such variation. Here on the right, you can see the PAM50 panel. It is a panel of 50 genes that shows their expression. High expression is shown in green and low in red. Now, you can also notice that we can see patterns or profiles of gene expression. Using various combinations of genes, researchers started classifying breast cancer into several subtypes: luminal A, normal-like, luminal B, HER2-enriched, and basal-like. How can this molecular subtyping be used? Because many patients characterized with such methods were observed for a long period of time, we can assess the risk levels for recurrence and survival. In this chart, you can see a Meyer-Kaplan curve of survival or recurrence based on two clinical gene panels, PAM50 and Oncotype DX. These panels separate patients into three groups: high, medium, and low risk. Based on these assessments, a treatment is selected. Traditional treatments for cancer are surgery, radiotherapy, and chemotherapy. Each one of these treatments can have a harmful side effect and is known to be effective in very specific situations. For example, tumors are typically irradiated before surgery or if they are considered to be somewhat benign. Chemotherapy targets cell proliferation. And often it ends up affecting many other cells in the body, not just the tumor, causing severe discomfort to patients and in some cases leading to complications. However, since the 1990s, a new type of treatment started emerging in clinical use: targeted cancer therapy. One such treatment is called Herceptin, or trastuzumab. It is a monoclonal antibody that blocks the HER2 receptor, blocking it from receiving the growth factor hormone and preventing cell growth. But even though Herceptin is considered a major breakthrough for patients with HER2 protein overexpression, targeted therapies have not become a silver bullet cure for cancer. In early-stage curable HER2-positive breast cancer, trastuzumab-containing regimens improved overall survival and disease-free survival. However, the studies also showed an increased risk of heart disease and resistance developed by most patients. Here you can see the proposed mechanism of action for this drug. As you can see on the picture, the antibody causes a sequence of events that disrupt the signaling and proliferation cellular pathway. On the other side, we see the disruption that takes place. These changes cause resistance or lack of response. Mutations, polymorphisms, and lack of expression are all factors contributing to this resistance. The study of how Herceptin works and its molecular mechanism of action continues, providing new insight into combinations of factors that could lead to a significant number of decisions that should be taken when prescribing the drug. From observed changes in receptor gene variation, ERBB2 encodes for the HER2 receptor, changes to other players in the signaling pathways, and other variation caused by the treatment response. This is a great example of how the complexity of subcellular mechanisms lead to both breakthrough therapies and new challenges, where each new challenge is characterized by many players and interactions between them. The study of these changes will be the focus of this project, where we'll take the gene expression data and apply machine learning to help us extract meaningful insight into the nature of breast cancer itself. The publication we are taking data from has over 50 different breast cancer subtypes. So approximately 10,000 genes are expressed for each cell line that has an associated clinical subtype. The same cell line were also treated with 70 different approved and experimental compounds, and IG50, or inhibition of growth profile, for each cell line was measured. The first thing that we would want to do is find this project on NCBI. So let's go ahead and do that. On NCBI, data is organized in bio projects that contain links to publications, various metadata about samples, and if raw data is available, it can be downloaded. Here's how you find the dataset and download information about the samples. Let's take a look at the downloaded table and see what's inside. Data is stored in a compressed format, or SRA, Sequence Read Archive. The SRA archive contains runs, or samples. They start with the letters SRR, or Sequence Read Run. If you download and extract the SRA archive, you will find that they contain FASTQ files inside, with extensions _1 and _2 at the end. Looking at the metadata given with this project, we can see that the data was produced using pair-end sequencing. It is a technique that uses simultaneous reading of each fragment from both ends, thus improving the quality of sequencing. In addition to the file name and its download location, the metadata shows cell line names used in each run and the clinical subtype associated with that sample. To save us some time, we already downloaded and unpacked the archive, preparing it for us to use on the platform. So for our first pipeline, we will select some normal-like and cloud and low samples to do the analysis. We have 5 normal-like and 6 cloud and low samples. You can do your own search on the internet and read more about these cell lines. The cells even look differently, as you can see in the picture. What we also know about the clinical classification is that cloud and low is a much more aggressive cancer that is often grouped under triple-negative breast cancer. Triple-negative means the tissue tests negative for 3 main receptors: ER, or estrogen receptor, PR, progesterone receptor, and HER2, human epidermal growth factor receptor 2. I think that we now know what kind of data we will be dealing with. As I said in the beginning, we have to carefully prepare the data for any machine learning. In the next session, we will take the uploaded files on the tBioInfo server and run an RNA-seq pipeline to generate a table of expression.